So I think we should move swiftly on to the next presentation. We've got a double act here. So if I could briefly introduce firstly Dr. Bhuvaneish Selvaraj, who's a postdoctoral researcher at the Ewan MacDonald Centre, University of Edinburgh. Most recently, he's led some research investigating one of the commonest mutations in ALS, I think highlighted in the earlier presentations. And this has led to a high-impact publication in Nature Communications earlier this year. So congratulations on your success there. I think Bhuvaneish is going to lead off, and he'll pass the baton to Dr. Arpan Mehta, who graduated in medicine from the universities of Oxford and Cambridge. He's currently undertaking his PhD at the Anne Rolling Regenerative Neurology Clinic in Edinburgh, and his research is funded by, amongst others, the MRC and the MND Association. So thanks to you both for your presentation.
So good afternoon, everyone. So I'm Bhuvaneish Selvaraj, and along with my colleague Arpan Mehta, what we are going to do today is going to give a little bit more insights into how stem cells, as such, can be used in the in motor neuron disease. So we are part of a large group of research organization called Ewan MacDonald Centre, which there are about 200 researchers, along with Anne Rolling Clinic, who are mostly widespread across Scotland. We are mostly interested in working on the motor neuron disease and bringing benefits to the patients. So, as everyone knows, there's a problem as such. I'm not going to explain more about the motor neuron disease as it's been explained very well by Brian Dickey and Pam Shaw. So I'm just going to tell about the problems, what we all know actually. So the neurodegenerative diseases as an umbrella contains quite a lot of like Alzheimer's disease, Parkinson's, ALS, motor neuron disease. They all are progressive and debilitating disorders. And this is caused by, as you know, loss of neurons and the supporting cells of the brain, which are the astrocytes, or we call it the glial cells. It is terrible and very devastating disease, and the prognosis, as you all know, is very poor. And the worst of all is we don't have any more effective disease-modifying treatments. And if you rank a list of illness which succumbs to death, and for that which we don't have any more cure or any more treatment, all these neurodegenerative diseases, what I said before, are ranking in the top 10. So that tells something. There's quite a lot needs to be done to overcome that challenge. And as you all know, it's a major economic burden to individual and the society, economically and financially. So there is, and as a gentleman pointed out, why we haven't done anything in the past 30 years. Apart from many other reasons, there are one particular reason. One is we've been access to the disease material. So for example, so if you want to study any disease, we wanted a live human material. For instance, in cancer, it's much easier, or any other blood disorders, you can take the real life blood and test them. But that can't be done in brain disorders, especially with neurodegenerative conditions. So you can't put a needle and take some biopsies and test them in the lab. So we were lacking material to do any live human-based research. And secondly, what that led to is we started doing quite a lot of animal modeling using rodents and Drosophilas, which is brilliant, but there's a huge translation leap between humans and rodents because there's quite a lot of evolutionary divergence. So that lends quite well why we don't. So that answers a part of the problem why we haven't found an effective treatment in the past 30, 40 years with the help in neurodegenerative diseases. So what can we do with the stem cells? So as we all know, stem cells are building blocks of the brain, of the body. So then the main, so this is a pointer. So these are embryonic stem cells over here, So and what it can do is the power of stem cells is it can develop into any cell type, any organ, and so therefore the entire human body. One important thing is broadly there are two types of stem cells. One we call it as a pluripotent stem cells, which can form, as I said before, any different type of cells. And the other one is the adult stem cells. So these are specialized stem cells which are located in different parts of the body, but they can only proliferate or generate a niche type of cells. So for example, if you take a bone marrow stem cells, it can generate only subtypes of cells which are related to the blood. But the pluripotent stem cells, which are shown over here in the green, which is a colony of cells, so they're individual cells. There are probably about 1 million cells in that colony, and each cell can be converted into, and each single stem cell can be converted into many different types of cells, and it has a potential to self-renew as well, so it can proliferate on its own, so we can cryopreserve. So what it gives us an opportunity is you can scale up quite a lot. But again, the embryonic stem cell, how do we get that? So it was isolated in late 90s during from human in vitro fertilization before pre-implantation. So you can take few of the cells, and those are the cells which are embryonic stem cells, which are pluripotent, which can again be converted into many different types. However, again, there's a problem over there. If you are studying a disease like a neurodegenerative diseases, which are coming at pretty late on side, so we don't have access to this embryonic stem cells, because those are only during the in vitro fertilization, and there's no way we can get that. So that's where the new technologies are very important. So the one important question is, can we turn back the clock? So meaning, can we turn the adult cell, which are like skin cells or the blood cells, back into stem cells? Therefore, we can have an unlimited supply of patient-derived cells, and we can model them into neurons or different parts of the brain, and then do a disease modeling. So one of those pioneering work was done in Scotland, which you all know is one of the highly celebrated sheep in the world, which is the Dolly. So that's the first example of cloning, mammalian cloning using adult somatic cells. So meaning that they've taken a differentiated cell which have already had a postcode and taken that and then made a new organism out of it. Although this wasn't efficient, what it told us was there is a potential that if you can convert a differentiated post-mitotic cell back into stem cell, and therefore you can convert them back into whatever cell types we want. So that was in 1998, the Dolly the sheep from Sir Ian Wilmot. Fast-paced, 10 years later, there was an amazing Japanese scientist. What he did was he identified a trick whereby he can take any type of cells, like skin, like what Pam Shaw explained, and then added 4 different proteins together, and then he turned them into stem cells. So this process roughly takes about 4 months in lab. But what it essentially does is you can you can make stem cells from any patient at any time point. Nowadays, there are even opportunities to make stem cells from urine samples. So even if you're thinking about making stem cells from babies, you don't even have to do a skin puncture. You can collect the urine, take cells from that, and again reprogram them back to stem cells. So the advantage again here is it's now has given us we can make a bespoke patient-derived cells, and therefore we can study the human disease in a human system. So once we make all the stem cells, why is it very important? So this is a graph from one of our scientists, Ricardo Dolmes in Novartis. What it predicts is, what it plots is different models and how it is relevant to the human and how is it good for mechanistic understanding. As we all know, in our sciences, most of the study starts with human postmortem tissue. So once we know where the problem starts, then we trace back and try to find the mechanism. So the human postmortem is probably the starting point and has a higher human relevance. However, that doesn't lend too much into it to understand why is that happened. So it's always like the last point. Then we go into, when we start putting the same risk genes and into lower animal models and mouse models, and then they are brilliant for mechanistic understanding. What we can do over there is we can follow up from the start of the journey till the end of the journey and assess where things go wrong. They are brilliant. But the problem, again, is it is not high throughput, so it's laborious, it's time consuming. However, and again, there are quite a lot of challenges, as I said before, genetically and to translate anything from mouse and other organisms towards the humans. So that's where we think that iPSCs, or induced pluripotent stem cells, can lend itself that it can be more mechanistic, it can be higher of human relevance because it is from the same patient carrying the same genetic disorder. Second, it's got we can understand mechanistically because it is, you can make those cells grow in the lab, you can monitor them day in and day out, and you can find what the problem is. And thirdly, because it is unlimited supply, because we can make them in the lab in a dish, you can scale it up in a large, in a high throughput way, and then screen for quite a lot of compounds, drugs and stuff to reverse any mutation. So again, this is sort of an overview of what generally we do with the stem cells with respect to the neurodegenerative diseases. So basically, we start from the patient, take a skin biopsy or a blood sample, collect the cells, add the 4 proteins which is labeled over here, and then make them into patient-specific iPS cells. So from there on, if there's a genetic predisposition, what we generally, nowadays, what we also do is we do the gene correction using a latest technology called CRISPR-Cas9. So now what happens is now you have a patient-derived stem cells with a mutation, and then you have a gene-corrected stem cells as well. Now you can compare apples and apples, rest everything is the same apart from the genetic mutation. And then you can take it into 2 routes. One, you can have a cell therapy, which is quite popular in Parkinson's diseases, where you can make those, do the gene correction and make the cell type which is affected in the Parkinson's, which is the dopaminergic neurons. You can make them in a dish and then transplant it back to the patient. So that's one approach. That probably works for Parkinson's disease because that's only one cell type which is affected. However, for motor neuron disease and Alzheimer's disease, what is more pragmatic is what we can use these stem cells to make motor neurons or any affected cell types like other supporting cells in brain, and screen for and do a mechanistic understanding what is going wrong. And if you find a phenotype, if one of them is shorter and one of them is longer, which Arpan will follow up with after my talk, you can do a high throughput drug screening to find some drugs which can reverse any of those deficiencies. And then you can get that drugs back to patient. So I just wanted to end over here saying that this is one of the quotes from a Nobel laureate saying that progress in science depends on the new techniques, new discoveries, and then new ideas, and probably in that order. The reason why I'm reiterating is we started to get the tools ready for investigating most of the complex disorders in the brain. And I'll hand on to Arpan Mehta, who will discuss more about what we are doing in the lab with these things. Thank you.
Good afternoon, everyone. It's a great pleasure and privilege to be here. I'm doing a PhD with Professor Chandran, and Bhuvaneish is my bench supervisor, so I learn a lot from him. And he set the scene very nicely about what I'm going to try and wow you with, hopefully, which is what we're doing in the lab and some of the applications about what you've just heard about this amazing stem cell technology. So it all starts off with the patient and comes back to the patient. So you've heard very nicely that we can take a simple skin biopsy and reprogram these cells into stem cells, and then those stem cells can be given a postcode and made into any type of cell that you want. Now, we're interested, of course, in the brain cells, of which there are 4 main types. I'm very interested in the motor neuron, and as Professor Dame Pamela Shaw showed you, there can be the cable, the wire can be as long as a meter. That cable is called the axon. And however, that cell in its isolation is not enough to understand. We're understanding more and more in motor neuron disease that the other cells are as important. And so the beauty about stem cell technology is that you don't have to just study one cell in isolation. You can study them together. And so we're also able to make the other cell types, which are called the glia, or glue, and they're supporting cells. So there's the oligodendrocyte, which is the key cell in multiple sclerosis. And what that provides is the insulation around those wires of the neuron. You've got the astrocyte, the star-shaped cell, which was also introduced earlier. And really, really quite special because we're only just beginning to really understand the role of this cell in motor neuron diseases, the microglia. So these, I like to think of them as the brain police, and we'll see why in a moment. So by studying these cells in a dish from patients, we can better understand what's going wrong, and that's disease modeling. And if we understand what's going wrong, we can then try and fix it. The other nice thing about stem cell technology is that you can do very large-scale drug discovery. And this can be sort of two broad approaches. The first is you can use the information from what's going wrong to develop a new drug. That's quite hard. What's perhaps a bit easier, at least sort of conceptually, is to use drugs that are out there already and repurpose them for motor neuron disease, because there may well be drugs or even chemicals from nature which might actually have an effect, at least in the cell in the dish. And then, of course, it needs to be translated further to real-life clinical trials. So stem cells can tackle both of these things. And so, as I've already said, I'm interested in the motor neuron in my PhD. I'm sort of midway through. And I'm particularly interested in those long wires, the axon. And essentially what we can do, using microscopes and other technologies, we can look at the various components, so not only the genes, but the proteins, the cellular machinery, and ask what's going wrong. So I'm going to give you a flavor, and this is, stem cell research is a team effort. The stem cells don't give you a single day off, so whilst I'm here, my other colleagues are in the lab feeding the cells. They're very needy cells and very expensive to maintain. The first finding quite early on in my PhD is that, so I'm studying the C9orf72 mutation, which we've already heard is the commonest known genetic mutation in ALS. And what we see here are some motor neurons. They're colored in green. And on the left, you can see motor neurons from healthy people. And you can see the axon, the wire, is quite long. However, in the disease case on the right, it's much, much shorter. And these are from iPS-derived motor neurons. And so it's an interesting question why this is happening, and I'm actively looking at that. But if you could find a drug that converted that sort of lengthened the axon, made it back towards the left-hand side panel, that would be potentially a very important drug. And that's sort of more scientifically shown on the graphs below. So if we can make the pink bars up to the blue, we might be in for something that might be very, very valuable. I'm also interested in the cellular batteries, which are the mitochondria, that are whizzing up and down, as Professor Dame Pamela Shaw said, whizzing up and down the axons. And in red here, you can see these mitochondria dancing up and down the axon. This is a live image, so the pictures are taken every few seconds, and you can see those microscopic organelles, the mitochondria, moving up and down. And you can ask the question, well, are these going wrong in motor neuron disease? Now, if you focus on the pink panel at the top, which is labeled healthy, you can see that in healthy people, those mitochondria, as you can see, are whizzing up and down at quite a fast rate. A lot of them are moving. Unfortunately, in this model, in the disease case, which is the lower right panel, you can see that the mitochondria are hardly moving at all. And actually, some of you may be thinking they look a bit odd as well. They look swollen, they look more circular-like. So again, if we can find a drug that makes the bottom panel more like the top, that might be of interest. This is another thing we've found, and others have found across the world. So there's a structure inside the cell, which is sort of akin to the Royal Mail sorting centre. And what this does is it packages things, quite busy at Christmas time, packages things, proteins, and sends them to the correct place. This is called the Golgi apparatus. And again, what we see when you do a 3D reconstruction, again from these stem cell-derived motor neurons, on the left, you can see that this apparatus, the Golgi apparatus, looks like that. And you might think, well, that's fine, all very, very interesting. But if you look at the right, the disease case, I think I can convince you that the Golgi apparatus look very swollen and fragmented. And you can ask the question again, why is this happening? But you could also say, well, are there drugs which can convert the right to the left? These are some really amazing videos from colleagues in our lab. On the left is the oligodendrocyte, which I showed you. We can make oligodendrocytes in a dish from patients, and this is exactly that. So I said the oligodendrocyte creates the myelin, the insulation, the sheath around the neuron. So what you can see here, really amazingly, is the oligodendrocyte there in the center, and the tubes, the sheaths, the myelin can remarkably be actually modeled in a dish. And then this is my favorite video on the right, the cellular police. So here you'll see the microglia. So there is this idea of the role of inflammation in motor neuron disease. And these cellular police, you'll see, they have these appendages. They go grab nastiness and gobble them up into the cell. And you'll see that we've put beads inside the dish, and when those beads are taken up by the cell, they fluoresce green. And you can actually count how hungry this cell is by counting the number of beads. And you could ask the question, are there fewer beads being taken up? So I'll just show you the video. So you can see the beads being engulfed by the microglia into the cell. So I'm just going to end, I've got a minute left, with the drug discovery part, and this is obviously very important to you. So it's a very long journey, far too long, to find drugs for this devastating disease. And so we need to accelerate this, and stem cells can help us, I believe. There's a 1 in 10,000 odds here in the classical pathway, and we need to do better. So if we can accelerate this process, it's a great thing. And I wanted to bring the dish with me, but basically you can grow these stem cells, these neurons, in a plate. So here there are 96 of these small wells, and each of those 96 wells you can grow hundreds of neurons. And then you can basically do a massive drug screen of drugs that are already out there and novel drugs, and ask the question, for instance, is there a drug that can make the axons bigger? And so I'm going to end with this slide, which is showing that this is really happening right now. So this is a video of one of these 96-well plates being taken out of an incubator where the cells are being looked after at 37 degrees, taken out automatically and put onto a microscope system. Each of those wells can have a different drug, for instance, and the microscope takes the images and produces the data. And then you use clever artificial intelligence software which can make those sorts of comparisons. So I'm going to end there by saying thank you very much. None of this work would be possible without your support. And we'll be very happy to take questions and meet some of you afterwards. Thank you.